Welcome to the lecture on protein prediction, and this is the first test whether you can all hear me, because if you don't, I currently don't have a mic. The easiest thing would be for those of you who can't hear me to just come closer. The less easy thing would be to go somewhere and get a mic. What do you prefer? Anybody trouble hearing me? Okay, so I assume that it's okay for you. We apologize that we had to move, but we were overwhelmed by the amount of students who registered for our lecture. So it's great to see that so many computer scientists are interested in bioinformatics, and we hope that we're going to meet your expectations. The lecture I'm going to give today is mainly a reminder on any chemistry, biochemistry you might have had at some point in the past. And it serves to bring you up to speed with all the terms we're going to use and we're kind of taking for granted when we talk about proteins. So to get an idea of what your background information is, I'd like to know who had chemistry in his last 2 years of school. Okay, at least a proportion of you did. Who has had some kind of chemistry or biochemistry in university? Okay, also a few of you have. So that's great, because I'm going to try and do this interactively. So I'm going to ask questions instead of just saying everything. So especially those who had something can try and answer. And the point of making this interactive is that you learn a lot more if you try to think while you hear me talking instead of just dozing off. Okay? Oh yeah, I haven't introduced myself. My name is Andrea Schafferhans. I'm a postdoc in the group of Burkhard Rost. I have studied chemistry and then via biochemistry at chemoinformatics I moved into bioinformatics. So I have never learned computer science formally in university, but the stuff I'm talking about today, I have studied that. So I hope I'm a good substitute for Burkhard Rost to teach you that. Okay, so what am I going to talk about today? One thing is just a general biology reminder. What are organisms? What are genes? What is the central dogma of molecular biology? And the other thing is we're going to go into proteins and chemistry. What are amino acids? What is protein structure? And also some physical chemistry about bonds and energies. Stuff that helps you understand how proteins work. And in the next lecture, which is not going to be this Thursday, but the Thursday after Easter, then Burkhard is going to tell you something about 3D comparisons about proteins, just to so you have an idea of what you can expect in the near future. Okay, so this is just to have a nice introduction and to give you an idea. All of these are life forms, and as you can see, life can be pretty diverse. There are some things that are common to all of these organisms. And the first question is, what is life? So if all of this is life, what is life? Sorry? A gene? Energy, yeah, something important. It's about overturning energy in a way. Other things, what makes life? Sorry? Growth, yeah, also an important thing. Other ideas for life? Information, yes. In order to grow and pass on, you have to pass on information. What is the difference between something living and a chair, for example? What makes matter live? Reproduction. Yeah, right. Growth and reproduction is two different things, right? Okay. So one thing they all have in common is they have some kind of organization. They usually have some unit cells. A virus has that, but also every cell in your body, every bacterium, etc., has got an organization form. You mentioned growth. Another important aspect of life is that it can adapt. It can adapt to its environment. It can respond to outside stimuli in some way. You had reproduction, then we had the turnover of energy or transfer of energy, and that usually occurs in some kind of metabolism. And you have something which we call homeostasis, which means that the cell can keep constant conditions even if the outside changes somehow. So the interesting question, which is still debated, is are viruses life? Yeah, well, it gets really difficult here. What is a virus? And why do you think no? Came from there somewhere.
Yeah, I don't really know why, but I'm very sure you know.
Okay, can somebody help him? Why could a virus not be life? What does it not have?
Homeostasis. It can be frozen, and it can be at a constant state for a long time.
Actually, you can freeze bacteria. Yes, but usually viruses do not have homeostasis. The interesting thing is, in biology, there's always an exception. And people have found a virus called mimivirus, which has got an amazing amount of things, which you usually only find in things like bacteria or other domains of life. It's got a cell membrane. It's got a kind of core, like the nucleus in bacteria, which usually viruses don't really have. And it even has some proteins that live inside this virus and keep up some kind of homeostasis, etc. So this is just to show you that any kind of lines we draw in biology are not really fixed. You know, there's always a gradient between things. But viruses tend to mainly consist of the genes they transfer and a few helper proteins, but don't really have life as such. Okay. Now, to the central dogma of molecular biology. So the genetics, or the information about how an organism, a cell, is made up, is encoded in the DNA. From there, it's copied to the RNA, which is kind of a transfer form for this information. So it's a bit like you have a hard disk where the program is written onto, and then you load this into the memory, and so you can execute the program. And the RNA is a bit like the form of the information loaded into the memory. But then from there, the information is used in order to build proteins. And mainly the proteins are those things that are really active in your cells. Okay. So basically, this is what I learned in school. This is also what I learned when I was studying in the 90s. And again, gradually we're finding out it's not completely true. At the time when I learned it in school and I was studying, RNA was just some transfer form for information. Nowadays we're finding out that a lot is happening at this stage, and a lot of regulation, etc. is happening. But from your point of view, the main thing to keep in mind still is that there is a connection between DNA and proteins, and that the important thing is that the DNA encodes for how you build the protein. Okay, that's the thing to keep in mind. Oh, and oops. And you probably have all seen some pictures of DNA. Because I'm a chemist, I always like 3D, and that the molecules are not just some 2D graphs sketched on paper. This is like DNA looks in 3D. And the base pairs that people always talk about are here in the middle, and on the outside you see kind of backbone that carries the bases here. And this is just one form how a protein can look like. Okay, so why do we talk, I mean, in a lecture about protein prediction, why do we talk about DNA in the first place? Well, the thing is that it's pretty hard to determine the sequence or how a protein is made up just taking the protein. But it's pretty easy to work out which bases the DNA is composed of. And that is because we can use all the mechanisms that nature has in order to multiply DNA. So if you find a probe of DNA somewhere, you can use enzymes to make more of it. And you can also use enzymes to help and determine the order of those bases. So basically, DNA, how many bases are there in DNA? How many kinds of bits do we have in biology? In computer science, you have 1 and 0. What do we have in biology? Yeah, we have 4. And they are called cytosine, guanine, thymine, and adenine. And the order of finding those in the DNA makes up the information you get. And what you can do is you can, if you have a strand of DNA and you want to find out what are the bases in there, you can take enzymes and get them to copy this strand of DNA. You can put in a few bases where the copying machinery can't go on, so it has to stop. After it has put that one in, it has to stop. And now stochastically, it will sometimes put the stop thing in the first cytosine, and the second cytosine, and the third cytosine, etc. Depending on where it puts on the stop cytosine, you get a different length of DNA. Now, if you label those stop things, you have one label for cytosine, one for guanine, one for thymine, etc. So you label this with a coloring agent. Now you get different lengths of copied DNA, and each has got a label at its end where it stopped. What you can do now is to kind of sort the pieces of DNA by their length. You can do that in the lab by running it through a kind of gel, and the gel is like a filter, and the smaller the DNA piece is, the faster it is, the longer it is, the slower it is. And that works more or less linearly, so you can really easily sort the DNA by length. Now what you get is, you can look at what color do I see. First I see something black. So I had a very short piece of DNA which ended with something black, which means it ended in a guanine. Then I get a slightly longer piece of DNA which ended in a thymine. So I know the second one must be a thymine, etc. So that is how you can determine a DNA sequence. And because this is really easy and works very, very well, what we do in order to find out something about proteins is not to analyze the proteins, but we analyze the DNA. And then we work from the DNA, we work out what proteins are there. We can do that because of the central dogma. We know that DNA is translated to, transcribed to RNA is translated to proteins. Okay? So that's why in a lecture about protein prediction, we still talk about genes and DNA. Okay, so how does this translation work? Well, I guess most of you have seen this before. There's a code which says that if you have those bases after each other, this means translation into that kind of amino acid. And the way you read this codon wheel is you take the outside. This is the first the first codon, for example here, a T, then no, sorry, the wrong way around. You go from the inside, you take an A, then you take the second one, for example, a C, and then you can take any of those, T, C, A, or G, and you get a threonine as an amino acid. And what you can see is that very often the last position doesn't really matter. And that's, by the way, also called wobble position, because often the mechanism that reads the DNA at that point doesn't really have to really carefully read, because it doesn't matter anyway. But in some places it does matter. For example, here, if you have an A, a T, it does matter whether you have a TCA or a G, because the amino acids you get are different. But the main point is there is a way to translate the DNA information into protein. This is just to show you how this looks in oops. Sorry. Sorry. Shouldn't have included a film. Anyone know how to start a film? Sorry, I've got no clue how to start this in. Oh, no, it's working. Okay, so what you can see here is the DNA, which has been transcribed into RNA. Then we get a ribosome, which is the protein that is responsible for assembling the proteins. You see little carrier molecules called tRNA, which bring the amino acids to the ribosome. And here you have them again. And the little things attached here are the amino acids which are brought. So here you have a bigger view. This is an amino acid sitting on top of this carrier tRNA. And so the ribosome processes the RNA. Different tRNAs come, get attached, and on top here the amino acids get attached to each other to form the protein. So here's an inside view of here the tRNA attaches to the RNA, and again here you see the amino acids being attached to each other. And while the amino acids get added, the protein already starts to fold. Of course, this is just an animation. This is how we imagine things to be, and I'm going to say something about pictures and reality later in the talk. Okay. Just to give you an idea about the amount of information stored in the organisms. Meanwhile, we have sequenced quite a number of organisms, and more or less the smallest bacterium that we know, and that can maintain its own homeostasis and everything, is Mycoplasma genitalium, and it's got around 470 genes. One of the biggest number of genes we have found is the rice genome, with around 50,000 genes. So what do you think, how many genes we find in humans? Anybody, any ideas, or anybody has heard any numbers or anything? So do you think it should be less or more than rice? More than rice? How much more? Sorry? Okay. Do all of you think that humans should have more than rice? Sorry? Otherwise I wouldn't be asking like that, wouldn't I? Yes, so people at the point where we had all of those numbers, that was in 1999, people were thinking, okay, humans are so complex, we're sure they have more than rice. So they have 100,000, something like that. And then slowly and slowly we had to go down. Currently the estimate, I mean, interpreting the genomic sequence is still not really easy. So we can't really say once you have the DNA sequence you exactly know about the proteins. But currently we're estimating around 25,000 genes. So an interesting question would be, how can it be? I mean, humans are pretty complex. Why so few genes? Yeah?
Maybe that for rice there is many transformation of genes that doesn't affect the viability of the rice, and because humans are so complex that if you change a little bit the gene, you will give something which is not viable.
That's a very good idea. So one thing is that it does happen regularly that genes get copied, and then maybe both copies just work and they're carried on. Oops. Oh, shit. But the other thing is that we also, as I told you, one of the problems we have is that it's not that easy that from one gene you can go to one protein. There's lots of processing happening in between. And there's a lot of information also encoded in the regulation when you express the gene. Do you just read the DNA information as is, or do you cut out some parts and make different proteins out of the same gene? That is also part of the answer. So there's a lot of complexity hidden in the stuff where I said actually it's not as easy. But this is under constant research, and it's too early to really tell you lots about it. It just should make you aware that this is an area where biology is moving on, and you can expect to learn a lot more, which goes beyond the central dogma. Okay, so now that we know the DNA sequence, and we know the DNA sequences of lots of organisms, do we understand it? Do we know everything we need to know? Well, obviously, it's a bit like having all the pieces for a car. Or a bit like you get every good manual. You hardly ever find what you look for, and when you find it, it's very difficult to understand. And actually, this fact is kind of our motivation for further research into how proteins work, because we are trying to find out for all of those pieces, what do they actually do? And how to find this out is what we try to teach you in this lecture. Okay? Good. So the full picture for us is that you can go from DNA to protein But then from the protein, you want to find out what is the structure of the protein. And once you have that, it's easier to predict what is the function. Sometimes we have to do without the structure and directly try to go from sequence to function. But basically, that's what we're trying to do. And this part is going to be a very, very large part of this protein prediction lecture. Okay. I told you before that a lot of the complexity lies in this intermediate step, the RNA. And there have been research projects, for example, this Phantom project, which is trying to find out actually what is happening at the RNA level. Because there's lots of processing happening there. So, for example, they have been looking at, you can read the RNA from one end or the other end, and what's the relation between that? Does it always also contain some information and things like that? Oops. Okay. Again, you should remember, so your take-home message from this lecture is the central dogma, but you should keep in mind that it's more complicated. Then a point about what is the information we have. Okay, so there is a lot of DNA information. Then, as I said, we translate the DNA into RNA. There's some processing happening here. Then you get the protein. You can take a protein apart into domains. You can find out something about the 3D structure, and we can have annotations about function. So just to illustrate what is the amount of information we have. So we have the entire genomic DNA of about 180 organisms. In the database GenBank, there are more than 14 million genes. In the Trumbull database, there's sequence information about more than 50 million proteins. And the difference is because of this processing, when you go from DNA to proteins. If you look at structural or functional subunits, we know of about 70,000. The number of structures, so where we know the 3D fold of proteins, is currently around 90,000. But this is partly redundant because the same structure has been solved a number of times. And there are around 100,000 annotated entries, protein entries in the SwissProt database. By the way, it's worthwhile to remember some of the names of those databases. So SwissProt and Trumbull are databases we use a lot because they contain a lot of the information we have about proteins. So they're going to be mentioned very often. So you don't need to be able to, we're not going to ask you to name a protein database, but the other way around, passively, if we say SwissProt, it's good if you remember as the protein database where information about the protein sequence, annotations, etc. is stored. Okay? And similarly, PDB, which is, so if we talk about proteins, it just happens that we say PDB and we assume that you all know it's a structured database. So try to remember the name. Okay, just to illustrate the growth. So this is the database containing all protein sequences unannotated. And you see that there's a tremendous growth here. Currently we're in the 10 to the 6th region. If you look at SwissProt, which is the number of curated sequences where we have some annotation information, that's one order of magnitude less. And yes, you can also see we're not keeping up in annotating. And again, the fact that we're not keeping up annotating and manually curating is one of the reasons why we need bioinformatics to work out the information computationally and to be able to make sense of all those sequences. And again, about an order of magnitude lower is the amount of known structures. Okay. So what are proteins? Oops. Why is this not showing? Okay, I added this pretty late and apparently my PowerPoint crashed in between. Should be on the slides which I put on the web, were just some very nice pictures of proteins. Let's go for the important part. What is protein function? So, as I said, proteins are the things in your cells. Yeah. Okay, so as I said, proteins are the main actors in the organisms. They're responsible for defense. So, for example, antibodies are for defending you against diseases. They're responsible for giving a cell structure. An example is collagen, but there are all sorts of molecules that give the cell its structure. There are enzymes that are responsible for building up and breaking down. They are responsible for communication and signaling. Insulin is an example of a protein that's responsible for signaling. They do ligand binding and transport. Hemoglobin, for example, what does it transport? Sorry?
Oxygen.
Yeah, right, it transports the oxygen in our blood. And they're also responsible for storage. For example, ferritin is again in our blood and is responsible for storing iron. So this is the main categories of protein function. So proteins are the main actors in your cells. And in general, proteins are gene products, and you can also think of them as the machinery of your life. Just a nice overview of what proteins can look like. Actually, David Goodsell is a person who's a scientist and an artist. And he's really good at drawing very informative, but also very aesthetic pictures of molecules. And in the protein database, the database that collects structures, there is a page called Molecule of the Month. So if you sometimes want to learn something about proteins, they have once a month a nice text about some protein where a new structure has been determined, and they describe it in a very nice way, and usually the illustration is from David Goodsell, so I can really recommend that as a regular primer on biology. And this picture is to illustrate the difference in sizes you can have in proteins. So for example, myosin is a molecule you have in your muscles, which make you move. And just to compare the size and the length of a myosin molecule to, for example, an antibody, or this is the outer hull of a virus. So just to give you an idea of the diversity of proteins. Okay, this was about the protein function, and as we've seen before, the proteins are made up out of amino acids. So far, amino acid has just been a name for the building blocks of proteins for you. Now what I would like to teach you is to understand what an amino acid is, because we talk about them a lot. We will not expect you to be able to draw an amino acid if we tell you draw a tyrosine. We don't expect you to be able to do that. But on the other hand, it's worthwhile getting to know the amino acids, because we often talk about properties they have. And for example, you can divide them into some that are more or less hydrophobic. What do we have? The hydrophobic. And some that are more hydrophilic, so some that like water and some that don't like water, and things like that. So this is that's the reason why it makes sense to have a more closer look at the amino acids, and by I guess you will know some of them by name. Okay, this was just a first overview of amino acids, and to remind you, the connection between amino acid and DNA is this genetic code. Here it's written in a different way. You've got the first position here, second position there, third position there, but in the end it's the same as in the codon wheel. An interesting point Burkhardt looked at some point is, do the different kinds of living organisms we have use the amino acids in the same way? And he found out that largely they do. So he looked at archaea, prokaryotes, and eukaryotes. Do you know what that means? The different kingdoms of life? Okay. So if you look at how life developed, you can basically divide different types of organisms you have. One are archaea. Then there are those that do not have a nucleus. So the DNA is not really encapsulated in a specialized organoid. And they're the eukaryotes. There you have a nucleus. What are humans? Yeah, we are eukaryotes. We do have a nucleus. And a lot of bacteria are prokaryotes, but not all. Okay. And as you see, basically, so each letter stands for a type of amino acid, and the size of the letter shows you what percentage of this amino acid you find in any kind of protein from any of those organisms, and you see that the distribution is pretty the same. This is not true for codon usage. Here's a comparison of the codon usage from a human and an E. coli. And you see that, for example, for arginine, the humans use all the codons you can have for arginine more or less the same. There are two exceptions. In E. coli, there's a strong preference for one codon. We do not really know why this is the case. But in some biochemical experiments, it's important to take this into account, because the bacterium can express a protein a lot faster if the preferred codon is used in the DNA than if a less preferred one is used. We assume that this is because there more of this transfer RNA for this codon is present in an E. coli than for the other ones. So, just as a side note. Oh, and by the way, of course, the amino acids themselves are absolutely universal. So there's not one bacterium or anything that has a different set of amino acids in the first place. Then something about amino acid structure. Why are those things called amino acid? So what does the amine in amino acid mean? Sorry?
Ammonia?
Yeah, right. It's got to do, it's the same root as an ammonia. Do they have? No, they don't really have. What does ammonia look like? Can anyone tell me the formula for ammonia? Well, it's true, and as a chemist, we also like it to draw it like that. By the way, does it occur like this in an aqueous solution?
No.
What do you usually have? Yeah, right. You usually have another protein here. Right, so, and if you have anything, so we call this R for rest, and then an NH2, this is an amino function. So this is the amino in amino acid. And like with the ammonia, in aqueous solution, you usually have another H attached and have this charged. Okay, what's the acid? Why is it an amino acid? Sorry? So we've got this COOH. And why is this acidic? Sorry? Yeah, so we've got this OH here, and this oxygen draws the electrons pretty much to itself, so the hydrogen has to go away, the electrons stay here, and we have the H+. Yeah, that's why this thing is called amino acid. And so in general, any amino acid looks like that. You've got the central C. We call this C alpha, because we kind of count the carbons, and this one is the C alpha. And very often when you talk about protein structures, etc., you just look at the position at the C alpha. So remember, this thing in the middle here is the C alpha. Then you've got the amino function, you've got the acidic function, and you've got the rest, which is the side chain. Very often when we talk about proteins, we talk about residues. Strictly speaking, a residue is just this residue here, attached here. But very often we mean the whole amino acid is made up and or incorporated into the protein. The other thing I told you just a moment ago is that ammonia and the acid in aqueous solution would usually occur in the, at least partly in the ionic form. And for amino acids, if you look at how do they look in an aqueous solution at pH 6, for example, this is usually the ammonium, and this is the carboxylate form. Another one, isoleucine, the same. And some amino acids have another basic or acidic part, and then usually at neutral pH, this is also ionized. Okay, so just to make sure you understand why this all happens, and when we talk about bonding, etc., some physical chemistry. What is a covalent bond? Well, a covalent bond occurs when you have two atoms. Here I've taken the simplest atom you can have, which is just a hydrogen. You've got the proton and one electron. The two of them come together and they share their electrons. So, and in chemistry, we usually find that thinking about probabilities of finding electrons somewhere are a good way of thinking of how molecules are made up. These localized probability distribution functions are also called orbitals. So when two of those things come together, they form an orbital which goes around the whole molecule and which has got a considerable probability of finding the electrons in the middle. That's what makes the bond between them. You can also look at the energy that a single electron, that two atoms experience when they come together. First, there's a certain attraction because the electrons are also attracted to the other nucleus. And then if the atoms come even closer, there's a repulsion because the nuclei also come too close. Some things are not just bonded by just one bond, but by a double bond. For example, here I've drawn a double bond. What's the difference between a double bond? Well, you've got, if you think about orbitals, you've got another molecular orbital which links the two atoms together. And there you have a high probability of finding the electrons above and below the plane. This is called a pi bond. And pi is pretty often also mentioned in other contexts, therefore it's worthwhile remembering that this is how a pi bond looks like, and this is what we mean when we talk about pi. The bond energy of a double bond is higher than just of a single bond. And another thing why this is relevant to know whether it's a single bond or a double bond is that you can't easily rotate. Because if you rotated this, then the two atomic orbitals wouldn't overlap properly anymore, and you would break the double bond. Okay, so that's an important thing to remember. If you have something that is bonded with a double bond, you can't just rotate. Okay, another thing to remember is that it's not always easy to draw how the molecule looks like, especially if you draw those electron pairs here. You can't really decide whether this is correct or this is correct. And you have to keep in mind that basically all of these orbitals overlap, and the structure is somewhere in between. And this also means that this thing is planar and pretty rigid and can't just move around those bonds either. I'm telling you this because it is relevant for peptide bonds, because they are planar too, and you have to keep that in mind. Another prominent example of delocalization and having electrons that can just move about is in benzene, where the whole ring in benzene forms orbitals that make the benzene planar. And again, you've got probability of finding electrons mainly above and below the ring. And now these orbitals from different benzenes can again interact with each other and form non-bonding interactions. So here I've been talking basically about bonds just as if both atoms were equivalent or the same. You should keep in mind that very often you have bonds between different types of atoms, and then often one atom can draw the electrons more to itself than the other one. This was relevant for this acid formation, for example, and in water, for example, this is the reason why water is a dipole. So you often have polar bonds. The other kind of bond you often find in nature is an ionic interaction. So one thing is charged, the other one is charged. The two charges attract each other if they're opposite. You can describe this by a Coulomb potential. And in reality, most chemical bonds are somewhere in between. You very rarely have only ionic or only covalent, but it's usually somewhere in between. Apart from real bonds, which are pretty strong, we also have non-bonded interactions, and they're extremely important for proteins because they make up a lot of how proteins interact and how they build structure. So the bonding amino acids together is done with covalent bonds, but within this chain of amino acids, the interactions are non-bonded interactions. And the interactions you have here is the so-called Lennard-Jones potential, which means that if two atoms come closer, again, there is an attractive force and the repulsive force, pretty much like in the bonded case, but the energies are a lot lower. And the terms describing this are different. To give you an idea of the strength of interactions, so this is what just Lennard-Jones would do, for example. This is in the order of 2 kilojoule per mole, whereas if you look at the gas phase, an ionic interaction would make around 400 kilojoule per mole, just to get you an idea of the order of magnitude. Of course, usually proteins are not in the gas phase, but in water, and there you've got the dipole of water, which shields the ionic interaction, and so an ionic interaction in water is a lot lower than this one. Okay, this is also to give you an idea of strength of bonds. I come to the hydrogen, yeah, so this shows you a hydrogen bond, which is also somewhat in between, because the oxygen of one hydrogen can share part of its electrons with the hydrogen of another water. And you can find that a lot in proteins, and this is what makes up some of the protein structures you'll see in a moment. This is a case where you have ionic interactions in proteins. You can have salt bridges. That means you have an acidic group and a basic group which attract each other. And on top of that, you can sometimes even have hydrogen bonds. And this is just an example where a salt bridge, or where salt bridges occur in real proteins. So just to show you, this is something real to look for and something real to remember. If you want to, for example, calculate the structure of a protein, if you want to predict that, because this is what this lecture is about, how to predict stuff, then you need to know this kind of interaction could occur and to look out for, do I have this here? Can this stabilize my protein structure? Another thing is, as I told you, you can have those aromatic rings, which give got electron density below and above, and they can stack together. So if two of them come together, a very favorable energy for them is if you can stack them together. And this stacking of aromatic rings is also something that stabilizes DNA, for example, because there you also have aromatic rings and you've got these stacked bases. And here again we have a 3D structure where you can see that these aromatic rings have oriented such that their planes are parallel to each other. Okay, so to sum up, what are non-bonding interactions you should remember? There are hydrogen bonds, there are salt bridges. I haven't gone into details for metal complexes. Usually a metal will also like to be close to some electron pairs from something else. And there's also something called cation-pi interaction. So again, if you've got this aromatic ring with electron density above, and you have an ion, then the ion is going to look for electrons on the top of this plane. Okay, coming back to proteins. We had the amino acids, and now we can look at what kinds of amino acids can have what kinds of interactions because of their side chains. Well, one thing is you've got side chains that have base groups here. Here's an amino group in the lysine. Here's a group in the arginine, and both can be positively charged. You've got acidic side chains, and the names of the amino acids tell you that because it's aspartic acid and glutamic acid. They're often negatively charged. You have some that are polarized because, for example, here you have an OH. So again, you've got the oxygen that draws a pH that is more positive. Threonine has got an OH, serine has got an OH, and glutamine and asparagine have a CO and an NH2, which is also polar. Okay, now as I said, the amino acids are the building blocks. Several amino acids together can form bonds, and the way it works is that the NH2 group of one amino acid displaces the OH from the other amino acid, and you get the peptide bond. And of course, this can happen again and again, and then you get a chain like this, which is a peptide. And at one end, there's an amino group left over, and at the other end, there's a carboxylic acid group left over. Therefore, we call this one end N-terminus and the other end C-terminus. And often when we look at proteins, we orient ourselves by saying, okay, this is the N-terminus, this is the C-terminus. And when we write down a sequence of which amino acid comes after which amino acid, we always begin at the N-terminus and end at the C-terminus. Okay. As I said, it's relevant to consider something like double bonds, because you can also write this in a different form. You can consider that the oxygen draws the electrons to itself, and some of the electron density goes over here. You can also consider this in terms of the orbitals. Then you've got some electron density below and above. The important thing is this is planar. This part is planar. So this subunit, which you can see here and here and here, is always planar. So there's when you consider how this can look in 3D, you can always think of planes that can just turn to each other. But the other parts in here can't be turned. Yeah, as I said, when we want to write down the information about the protein, what we often store in databases is just the one-letter codes of the amino acids, and this would describe a protein. Now, the sequence of amino acid types is called the primary structure of a protein. The way it folds up locally is called the secondary structure of a protein. Then you can have pieces of secondary structure which are oriented with respect to each other, and they make up the tertiary structure. And finally, different chains of protein, of amino acids can come together to build an overall unit, and this is called the quaternary structure. This is also a basic principle, and when we talk about proteins, we often talk about secondary structure or tertiary structure. So these are key words to keep in mind. So the basic secondary structure elements, I brought one here. So this is an alpha helix. And if you like, you can pass it through and try at least to spot the C terminus and the N terminus of this. So here in the graphics, you have an N terminus here, a C terminus here, and you see that the alpha helix always makes hydrogen bonds between the CO group from one amino acid and the NH group from an amino acid 4 amino acids away. So that's the general structure of this alpha helix. And unfortunately, we don't have a lot of these blocks for building amino acids, but it's very instructive to take something like that and build it up, because you notice how flexible it is in the beginning, and then when you make the hydrogen bonds, it becomes something really rigid. So you get a really rigid structure in the middle, and it's good to feel that, really. And around there, so all the residues, all these R groups stick out on the outside, and they are still flexible. And if you hold this in your hand, you will also notice that it's flexible outside. Very often you also notice that you can have sides of a helix that have properties. For example, if these are outside and stick into the water, then you will have one side of the alpha helix which is very water-loving, so hydrophilic, and you might have another side of the helix which is very hydrophobic. And I actually tried to build that in there. I tried to build a hydrophilic and a hydrophobic side in that helix. I had to stop building because we didn't have any building blocks left over. The other general principle which you very often find in proteins is the so-called beta sheet. This is because the amino acids basically form just a plane, and you get hydrogen bonds between one part of the chain and the next part of the chain, which comes after a turn. So when you load up the coordinates of where the atoms are from, for example, a PDB file, that usually contains the names of the atoms, where they are, and what the bonds are, and then it first looks like this, which doesn't tell you a lot, and you don't see much. What you can then do is to just show the trace of the C alphas. Remember the C alphas, where the central group where everything is attached to. Then you see more like, okay, where does the amino acid chain go through my protein? Technology is really not working here. I need a new computer. So where was I? So now I've highlighted parts of the structure. So can you tell me what the red stuff is? Yeah, red is alpha helices, and the yellow stuff is beta sheet. And so this is what it looks like in more detail. I told you about the properties of the peptide bond, that this is always planar, so you only have two bonds you can basically rotate around. We call this phi and psi, and depending on the angles you have, or depending on how you orient that, you basically get alpha helix or beta sheet. So if you just plot what kind of angles do I find in my protein at that point, you can also say, okay, this belongs to an alpha helix or a beta sheet. And Ramachandran made an analysis of what kind of angles can you find. And as I said, very often when we look at structures, we don't look at the atomic coordinates, but we look at more cartoon-like graphics in order to first work out how does this thing assemble. I'll soon stop. I just want to go through this protein structure, or finish this protein structure part. Okay, so to remind you, we've got the primary structure, secondary structure, tertiary structure, quaternary structure. I talked about the secondary structure. Tertiary is just how the different parts of secondary structure assemble. Quaternary is how different chains assemble. And we sometimes also mention disulfide bridges, which is a covalent bond, which often serves in order to stabilize the arrangement of the protein chain. So after all of the protein has folded, you form the covalent bond, and that helps to stabilize the protein structure. Okay. I had been planning to talk about this too, but due to all the technical problems, I think we'll shift that to Burkhardt's next lecture. Thank you.